Thank you very much for the nice introduction, and also thank you for the opportunity to talk about my lab's work. I moved to Memorial Sloan Kettering 5 years ago. I'm going to be talking about two projects today. One of them is ongoing at the University of Toronto, and one of them is in Memorial Sloan Kettering, so I'm reporting both of my affiliations. You might notice that the title is different. A lot changes in 6 months when you have to give the titles. I have a 6-month-old daughter. She chose last night to stay up all night. Okay, so I might be a little bit slow. So, broadly speaking, we're interested in, what I was told would advance the slides to not work, so let's try that. Great, okay. So broadly speaking, we're interested in cancer genomics, so cancer genome sequencing, identify somatic mutations. We're going to be largely talking today about sequencing bulk cancer populations for the reasons that a lot of this is going to have a clinical focus, and that's the clinical standard right now. As you know, you sequence a heterogeneous population of cancer cells. After you sequence them, do mapping and mutation calling, you can map them back to the genomes. And we're going to be focusing primarily on single base substitutions, but there'll be some copy number aberrations. And so I'm going to be talking about two projects by two fantastic graduate students, both of whom are here today, one of whom is going to graduate in a month, and and the other one is going to graduate shortly, and I'm sure they're both looking for their next role. And I don't know if Kate, do you have your project? Do you have a poster today? Okay, all right. They're both giving posters today. And so the first topic I'm going to be talking about is clinical deployment of tumor type classification. And that's what I originally wrote the abstract on until I realized I had twice the time that I thought I did. And so that allows me to talk about second project I'm also very excited about. And this is a new way of representing cancer mutation signatures that gives some sort of mechanistic insight. Okay, so the first project, this is work of Madison. And so why do we care about this problem? We care about this problem because about 3% of cancers, at least more than 3% of metastasis, are cancers of unknown primary, meaning that the patient presents to the hospital with a metastasis and it's not clear what the actual cancer type they have is. And treatment that's informed by the primary, generally speaking, performs better. Most of the targeted therapies require FDA approval requires that you know what the primary is. And for almost a decade now, Memorial Sloan Kettering is quite famous because they have this clinical genomic sequencing assay that they deployed in 2015. And so this assay is given routinely to patients. Initially, it was funded by a very generous donation, private donation, but now it's at the point that I believe it can be charged to insurance. And what this sequencing assay does, this is a targeted sequencing assay, meaning that it sequences about 1 to 2 megabases of the genome. These are targeted cancer gene panels. They target exons. And it's part of a clinical pipeline in the sense that all the mutations that are called have to be signed out by a pathologist. And the clinical report is generated that the pathologist looks at to make some reports on. Okay, and so previously, this is joint work with Mike Berger, who is the one who developed the MSK Impact panel, and he's the co-supervisor of Madison. And so my lab had previously developed a tumor type classifier called Deep Tumor, which was based on whole genome sequencing, and it was trained on data from 3,000 tumors as part of the PCOG consortium, which is a consortium consisting of people from ICGC and TCGA. And it achieved a relatively high accuracy. So it was a 91% accuracy on 24 tumor types, which is kind of a good point to be at if you want to deploy something in the clinic. And this was published about 4 years ago. In parallel, Mike Berger's lab, along with a variety of other labs at Memorial Sloan Kettering, developed something they called the genome GDENS or GDD random forest. So they trained a random forest classifier on data from 8,000 MSK Impact patients who had consented to research on their data, and it achieved 74% accuracy on distinguishing between 22 different types of cancers. Yeah, I'm a little awkward and uncomfortable. I'm not normally like this. I think it's because I'm tired. Okay, so The nice thing about deep tumors is the high degree of accuracy that it achieved. The bad thing about it is whole genome sequencing is not a clinical standard yet, and it's not clear when it's going to become a clinical standard. Whereas on the other hand, MSK-IMPACT is a clinical standard. It's been deployed in the clinic for a very long time, and there's cancer sequencing panels at a variety of different centers that are routinely given. So now what Madison then saw and set out to do was to try to make something that could achieve the classification accuracy of deep tumor, but on an assay that's routinely deployed in the clinic. And I should say one other thing is that we were very lucky to have the prior work done at Memorial Sloan Kettering so that we could essentially solve what I think of as a machine learning problem. So the random forest classifier from Mike Berger's lab, it was already integrated into the pathology report that the pathologists were looking at. A lot of the the questions on how to deliver the data to clinicians had already been worked out for us, though Madison obviously contributed more to that. And so our task was really to produce a better classifier. Now, one of the things that we had, my trick no longer works. Okay, now that trick works. Okay. So the thing that was different about deploying a classifier in a clinical setting is you don't get to control what your patient population is. You get the people who show up at your door, and the people who show up at Memorial Sloan Kettering, their cancer type distributions are extremely skewed. So there's about 2 orders of magnitude difference in the amount of patient data for the most common cancer types for patients in the cohort, which is a large proportion of the MSK patients, and the least common cancer types that Madison included in her classifier. And so that obviously, if you do machine learning, creates some difficulties, and because of massive class imbalance. And there's hundreds of more patients with rare cancers. There's dozens of more rare cancers that aren't even shown here. The framework that Madison chose to use, okay, I'm getting my air back. And so this is so what Madison called her classifier is the GDENS classifier. ENS meaning ensemble, and it's based on a hyperparameter ensemble of multi-layer perceptrons. And the training set that she used, it obviously increased in size from when the random forest classifier was trained, and now there was like 40,000 samples with cancer type labels. She chose 38 different cancer types. Then these cancer types collectively covered 97% of MSK patients. So it was an assay that could be broadly given to almost everyone coming into the door. And there's reasons for including the hyperparameter ensemble, which I'll talk about in a moment. And then the output of the tumor type classifier was both a probability distribution that gave the confidence on each of the 38 different tumor types, as well as through Shapley value analysis, a set of predictive features, which were the ones that were driving classification. Okay, and these are the input features, and you can see also the input features are very highly skewed. That's a log scale on the y-axis. The vast majority of them are hotspot mutations, and so I think Madison thought a lot about how she represented mutations. So she represented mutations at various different levels of abstraction. Are they hotspots? Are they mutations that are in the gene, for example? But importantly, there are a variety of other features, including both focal and arm level copy number variants. One thing to note, though, is the features are extremely sparse here. This is a targeted gene panel. It's only sequencing a small part of the genome. So the median patient has about 5 single base substitutions. Okay, and so as I already mentioned, the recognized cancer types, they cover 97% MSK patients. I want to highlight a couple things here. So the first is the cancers of unknown primary. This is this group right here. And I do not have, there we go. Laser pointer. I got it. Thank you, sir. Yeah, you can sit down. I appreciate it. It's very good. So these were not used as part of the training, but you can see they're about 3%. Also, these are the rarer cancers. So these are excluded types, and this is also about 3% of the cohort. Okay, and we'll get back to those later. This is the size of the training set for the various cancer types. Now I've switched to a log scale. So the fact that there's 2 orders of magnitude difference in size is a little bit harder to see. what Madison did to deal to a certain extent with the difference in size imbalances. She just upsampled the smaller categories. Okay. And to make a long story short, GDEMs perform extremely well. In fact, it performed as well as whole genome sequencing based classifier on the high confidence cases. And you can see the relative performance on all those things here on the side. So the high confidence cases are given by the dark colors, and the lower confidence cases are given by the lower colors. Blue means good, red means bad. So you can see for the kind of the rarer cancer types, even though we do very well on the high confidence classifications, we're actually missing a lot of them. And this is very much an area of future work, but we also have a very little training data for them. One of the reasons that you want to use a hyperparameter ensemble of multi-layer perceptrons is because it's this little hack, in the sense that in the clinical setting, you want your outputs to be very well calibrated. That means if you say there's a 75% probability that it's lung cancer, you want to be right, ideally 75% of the time. And so that calibration between your accuracy and your predicted accuracy can be measured with something called EEC. And it just turns out with hyperparameter ensembles, because each of the ensemble members is a little bit different than the other, they come out well calibrated without actually having to do anything else. Often with in other clinical settings, you'll do something called like PLAT calibration, and that becomes much more difficult in the setting that we're working, where you have to calibrate 38 different cancer types. Here we just got the calibration for free. Okay. And then the accuracy, as I said, was 93%, and that's for 72% of the patients that achieved that were above this high confidence cutoff. Overall accuracy was 80%, but we could identify which patients we were right about, which is important. The other thing, and this is something that the reviewers of the paper were very interested in, is it actually identifies the features that were important in driving the classification, and the ones where we knew what the important features were, like APC and colorectal cancer, it got right. UV damage caused by UV light and melanomas, it got it right. Overall, about half the time it used mutation features that contributed to about half of the signal, classification signal. The other half of the signal is CNAs. An important thing that Madison found is that the CNAs were the ones that were best for classifying the rare cancers. The rare cancers, okay, now we're talking about sort of more clinical things, and the rare cancers are actually pretty easily identified. So if I'd given this talk about 2 or 3 years ago, I spent a lot of time talking about how to identify out of distribution samples. It turns out if you are just looking at the most highly confident outputs, there's not really that much more that you can do. So luckily, about a third of the rare cancers were not high confidence. And for us, there's not a lot of clinical importance at looking at these ones. Then in the third that were high confidence, about 2/3 of those misclassifications, they got the organ system correct. So we don't know what rare cancer is because it's not represented among the 38 types, but we broadly know what the organ system is. Okay, the other thing which kind of became obvious very quickly in Madison's project is the problem that you're solving is not the problem that people are actually interested in you solving. So like, it's very rare that you don't know anything about the cancers that you're looking at. You at the very least know where the biopsy site was taken for metastasis. And because of organotropism, there are strong biases to certain sites for certain types of cancer. Often you also have histology. So the pathologist will look at the H&E slide and have at least some information about the type of the cell type of the cancer, squamous or adenomas. And so Madison, I think, this is one of my favorite parts of the project, is she had a very interesting idea about how to do that. And it's just coming up with an adaptable prior, meaning that taking the patient, the prior implied by the patient population that the classifier is trained on, and then changing it and adapting it to the prior that would be provided to you by a clinician. And in this case, we're actually automatically coming up with that new prior. So based on the biopsy site, the histology, and some information about clinical history. And so this is a real example. So you see along the top, this is the GDENS output, and this is the proportion, the cancer type proportion you would expect from a lung biopsy of a metastasis. And the third line here is what you expect from a squamous histology. And combining those all together gives you this final output, which actually is different with what GDENS would say, but is actually the correct classification in this case. Okay, and so now we get to talk about some math very briefly. So how does this adaptable prior work? So, so one of the other advantages of using a hyperparameter ensemble of multi-layer perceptrons turned cross entropy is you can interpret their output as an estimate of a probability distribution. In this particular, it's a conditional probability distribution conditioned on MSK impact. What's the cancer type? And what we'd like to do is we'd like to run GDENS, but we'd like it with a new prior that is implied by the data. Okay, and so this, by Bayes' rule, this is proportional to the probability of the DNA that you see, the profile you see given cancer type, and the prior on the cancer type. So in the GDENS, with the adaptive prior, we would like to change that prior in the cancer type. Well, we can easily do that from the equation above by simply taking the GDENS output, multiplying by the new prior, and dividing by the old prior. And so, and this new prior, you can derive it from other data just using naive Bayes. And so it's kind of a simple observation, but we're very excited about this because basically it means that you can add any clinical data you want afterwards in addition to information from other genomic classifiers without having to retrain anything. And this is known as late fusion. It's a particular type of late fusion. But it's nice to think about this as a set of modular tools that you can now put together. So let's say you have a classifier based on, say, gene expression, which is a very common thing to do for cancer type classification. Now, you don't often get gene expression data in the clinic, but if you were, you could easily combine that classifier by just using it to change the prior. Okay. And as a result of this, we saw a 2-fold to 4-fold increase in the number of cancers of unknown primary patients that could be matched to targeted therapy. And this is the high confidence predicted cancer types and the resulting matched therapy. And so one thing that the reviewers of our paper asked us to do was we said, well, we can now use this thing in routine use. So how useful is this classifier for the other gene panels? And there are a number of other major gene panels, and these are listed here. And so the good news is, if you just take the overlapping features out of the box with GDENS, and you just assume that none of the other genes have been observed from these other panels, and you rerun it on our cohort, you maintain a very high accuracy in classification, all the way down to like just 68% overlap in the genes that we see. And you still have a large proportion of high confidence patients, and you have high degree of accuracy in those high confidence patients. The bad news is very rarely do these panels actually report all the features that MSK impact reports. And some of these panels don't do normal sequencing, so that their mutations are polluted by normal ones as well. So this is what we could potentially do. The accuracy that you can achieve, at least out of the box, is a little bit lower. But if you retrain things, and I thought Madison was going to talk about that at her poster, and maybe now she might have to, things work much better. Okay, so in summary, so high accuracy tumor type classification is already possible using a routinely applied clinical test. And this test works for 97%, or is designed for 97% of MSK patients. And the 3% of the patients that have rare cancers, almost always we can identify them because they have low confidence classifications. And the other thing is that this works with cancers with purity as low as 10%. Okay. In some sense, it's also trivially multimodal via late fusion, using this adaptable prior mechanism that Madison invented. And it's important because this data can be incorporated without retraining. You can just take this as a module, apply it anywhere you want. Now, the caveats is that we need tumor normal sequencing right now, and we rely a lot on the MSK impact analysis pipeline. A lot of the other panels, they don't give us focal copy number or arm level copy number, and those features are incredibly important. Okay, switching gears And I'm going to talk about something that is like maybe a little bit translational, but maybe a little bit more translational than a normal mutation signature talk. Okay. And then this is work done by Kate Hartigan, and this is joint work with Tyler Funo at MSK. And we rely a lot on OICR for compute. And Kate is at the University of Toronto. Okay, so broadly speaking, my lab is interested in what I call reading the cancer fossil record. And we got interested in cancer because we were interested in cancer evolution. And generally speaking, people in cancer evolution think of it as a multi-stage, multi-phenotype process. You start from a healthy colon in the Vogelstein model of colorectal cancer. You have like a small polyp, a larger polyp, and then you have like carcinoma. The transition from each of these states is due to a single founder cell. And that transition ends up being important because that means that the population gains all the mutations that were present in that founder cell. And we know from our work on PCOG that more than half of the mutations that you see in the cancer are deposited before this transformation into carcinoma. So those mutations are indicative of one or more of these precancerous cell states. And one of the things we're broadly interested in is like modeling these fossil eras and then trying to understand what that tells us about the state of these precancerous cells. And so within the context of that, we think a lot about what the type of information you get from single base substitution mutations. So each mutation gives you a type. So that's the type of substitution that you see, where that mutation is, how common it is in the population, and mutations that are more common in the population happened earlier, and its function. And so largely we ignore function because 99.9% of mutations are not driver mutations. So they're either having no functional role because they're silent, or their functional role is not impacting or changing the cell state. And so one of the first things that people started determining that you could use these mutations for is saying something about the DNA damage repair processes which were affected in the cancer. So how does that work? Okay, so you can take the mutation, you can represent it as one of 96 different types. So there's 6 possible substitutions if you only look at the pyrimidine. And generally speaking, to understand the probability of a substitution within a cancer, you need to look at the 5' and 3' base for context. And if you take all that information, that gives you a 96 different mutation types. And if you just plot the histogram of those mutation types in different cancers, you get something that's very characteristic, often of cancers, but also of various features of the cancers. And so I'm assuming this audience doesn't know a lot about these mutational signatures, but these are just the histograms. And this thing here, these 4 lines, they're like the lifelines in palmistry. They tell you how old the person was that the tumor was taken from, because these are like these mutations, these 4 types of mutations, they accumulate like a clock, in kind of a clock-like fashion. This mutation right here, I hope that's not my phone. Good. All right, that would be embarrassing. This mutation pattern right here tells us that this person with the lung tumor is a smoker, because the CDA mutations are caused by tobacco mutagens. And then there's a mutation process called APOBEC that has occurred that's often in response to inflammation as part of the innate immunity system. And that's reflected by the presence of these 4 bars here. Okay, so you can take any one of these mutational spectra and you can break it down into contributions of that mutational spectra by different types of what are called mutation signatures. So I told you about smoking. So 52% of the mutations in this lung tumor were caused by the tobacco mutagens or associated with it. 25% of the mutations were associated with this APOBEC signature, this intrinsic cell process. There's some signatures of unknown etiology, but you can identify them as being entities by taking tens of thousands of tumors and looking for patterns of co-occurrence of mutations, which is how these signatures were defined in the first place. But the important thing here is you can measure these exposures, which is the proportion of mutations due to each of these types. And so one of the first things Kate did when she was actually an undergrad in the lab was to work on this method we call TrackSig, which tracks change in exposures of different mutation processes over time. And these changes of exposures are also very tumor type specific. So 70% of the time, the mutation signatures exposures change over the transformation of the cell from the normal cell into a cancerous cell. And in this case, for example, they're very cancer type specific because this mutation process, which is called SBS signature 9, is associated with the action of polymerase eta on the IgH locus in maturing B cells. And so this is a B cell cancer, and generally it's associated with a type of cancer called CLL. Okay, and you can see that these mutations are happening early clonal. This is before the transformation, because that's just the definition of the cell type. But as the cancer changes and progresses, you see more increase of this other type of mutation called SPS5. Problem with these signatures is they're not very interpretable. Here's some of the signatures from Cosmic, which is the database that contains all of these. And they do have a proposed etiology. But if you've tracked this field over time, the number of signatures seems to be growing at an exponential rate. And now there's tissue-specific signatures. There's signatures that are specific to different types of experimental perturbations. But it kind of seems that that's not the right way to think about these types of signatures, because they're reflections of a damage process and the repair of that damage. And there's just not that many repair processes in the cell. And so there's this really nice observation by Teresa P., who's here today, in this paper that was eventually published as Repair Sig, but we saw it at PSB, is that the damage and misrepair of mutation are separate processes. So the targeting, which tells you what base gets mutated or which base gets damaged, that's a damage process. Whereas the base that gets repaired, that's due to whatever the repair process that would affect that damage is, right? And there's different repair processes in the cell, and the relationship between damage and repair is not one-to-one, right? And so what these mutation signatures are actually doing is they're modeling a specific type of interaction between potentially multiple damage processes and multiple misrepair processes. And so what Kate sought to do was try to model these types of things to come up with a new way of representing mutation signatures. And so here's the statistical model. And if you like plates, then you can look at the plate model. Otherwise, I'll take you through it a little bit. Okay, and so what this model has is it has a set of so-called misrepair signatures. And here, just for identifiability, Kate represented just the substitution pattern. So there's 6 possible substitutions, and it's the distribution over those substitutions. And then the damage is the 32 possible contexts. So that's represented as distribution over that. And fitting this model to data from 19,000 whole genome sequences, she came up with stably about 18 damage signatures and 6 misrepair signatures, which is a much more efficient representation of this process than what is now in the hundreds of SBS mutation signatures. And so in the middle of this thing, and I think this is an important part of this, and this was also in repair SIGs, is the damage misrepair interaction matrix. So there's activity of a damage, so that tells you what proportion of mutations are due to each damage signature. And then this tells you for each damage signature, what proportion of the mutations are associated with an individual misrepair signature. Now these interaction matrices, they're estimated separately for every one of these tumors. Now, if you're like careful and fast in multiplication, you might be a little bit concerned about the fact that there's more elements in the interaction matrix than there are in the 96 different categories. And the way that Kate deals with that is with a sparsity prior. And it's a hierarchical model, so there's tissue-specific parameters of that sparsity prior. Some tissues have more interactions than others. Okay, all right. And so, the first thing is to sort of understand what's going on and what the different representations are of the damage versus misrepair. And the damage signatures, the first things you saw is they're much more tissue-specific in misrepair signatures, is what you would expect, right? Misrepair signatures are talking something about the intrinsic processes that are repairing DNA damage. You expect those to be present in every cell. You expect that to be a lot less tissue-specific, but the damage is very much cell-type specific. The other interesting and surprising is I told you that mutation signature changes in most cancers. Well, most of the time what's changing is the damage activity, not the misrepair activity. And again, that I think is a little bit, for me, is kind of expected, right? Some cancers you see a change in misrepair, but whenever you see a change in misrepair activity, it's because the damage also changes. So either there's been a change due to a new type of damage, or there's been a change in the efficiency of DNA damage repair, which reveals a type of damage that you didn't see before, right? But most of the time, it's damage that's changing when you go across cancer types. And you can look at this, and so for this reason, we think that this gives you a little closer view into the actual mechanistic features underlying this change in these mutational processes. So to look at this, she looked at some data from this paper from John M's lab at Memorial Sloan Kettering, where they did knockout of a variety of the enzymes that are involved in the ApoBec mutation signatures. And so there's two major damaging enzymes, ApoBec A and ApoBec B. And what she found is that, largely speaking, what's most affected by the knockdown is the change in the interaction profile between them, and there's a large change in the type of damage that's deposited. But not all the misrepair activities are changed. But I think the more another exciting result is that UNG, which is one of the enzymes that's involved in a specific type of damage repair, when you have loss of UNG or you have low expression of UNG, you see a change that moves away from misrepair 4, which is a specific type of misrepair activity, and you see much less change in the damage. So what's happening here is Kate's model is isolating changes in DNA damage efficiency from changes in the thing that's causing the damage itself, right? She looked more broadly, asking whether or not you could then use this model or this representation as a better way of highlighting what DNA damage processes are being affected in tumors. And she found that a feature set derived from Demuda, which was like a feature set of 24 elements, was much better at predicting which pathway was altered than a feature set derived from like 75 cosmic signatures. And in particular, the most important features for predicting which repair pathway was impacted by a loss of function mutation in the cancer were the misrepair features, not the damage features. And so, what you would obviously want to do then is to do the leave one on cross validation. Let's pretend we don't know a gene in the misrepair pathway. Can we recover it? Now, so one thing I should say is there's not a lot of genes in a lot of these pathways. So this is actually a hard classification task, but the features derived from Demuda are much better at this task than features that you derive from cosmic signatures. So just in closing, so Demuda has defined a new set of damage and misrepair signatures. We think this is not only a more efficient representation of SPS mutations, but also one that's easier to connect to the actual mechanisms that are being changed in the cancer. So I'd like to acknowledge the people who did the work in the lab. There seems to be a random highlight here because I stole the slide from somebody else. But this is my lab, as you can see quite nicely here. Our collaborators at Memorial Sloan Kettering, their major collaborator here is Mike Berger, and we had a lot of help from a variety of clinicians in interpreting the output of the model, but also understanding how to design the model. Demuda is done in collaboration with Tyler, who until recently was at MSK, and Karen Campbell, who is Kate's co-supervisor, and none of this work would have been possible without the help of OICR. Kate has a OICR appointment, and Michelle Brazos has really funded and kept my lab together since I moved to Toronto. And these are the agencies that funded this work. Thank you.
Is it on? Yeah. Really nice, Kate. Very nice. There's been a lot of progress since, when was it, last May? It's really great. Nice to see. So, did you get any insight into SBS5, for example? The flat signature that's clock-like, yeah. Sorry. Can you pass me the microphone?
Oh.
Yeah.
Yes, we have a little bit of sort of interesting, basically what I've seen so far is that there seems to be like a very consistent damage profile for that and some tissue-specific repair, and specifically CNS seems to be doing...
Which one?
CNS tumors seem to be quite different from a variety of other tumors in terms of their SBS5 repair response.
Right.
I don't know if that's just because those are mostly pediatric patients in our data set, but I think that there may be some interesting questions to keep asking about that in terms of the like sort of aging repair that we're seeing there.
That's really great. Thank you very much. Yeah.
Thanks. Both projects are super cool. I have a question about the first one. I saw you ordered which types of tumors you could predict better. And obviously there's going to be a correlation with sample size. But if you could control for sample size, specifically my question is asking, the thing you predicted the least well was gallbladder cancer. And I'm interested in gallbladder cancer. And is there anything that we can say about gallbladder cancer from it being poorly predicted? Like not comparing to maybe lung cancer where you got tons of samples, but comparing to the other ones with a similar sample size or something like that.
Madison, you want to answer that question?
My interpretation of gallbladder being one of the poor performers is because it's fairly similar to some of the other larger cancer types that are within our data set. So I think colorectal, it's frequently mispredicted as, and because colorectal makes up such a larger proportion of the cancer data or the data set that we have, it's just frequently mispredicted there. I think encouragingly, the high confidence predictions that we make are generally correct. So it's just how can we increase the proportion of gallbladder cancers that are predicted with high confidence? One way is to get more data. So hopefully like in time, the proportion of gallbladder cancers that we are able to include increases and we're able to generate more predictions there. And then any type of, I think to answer the question specific to gallbladder, I'd have to look at what kind of mutations are being implicated there. And also if there's any way, so right now we use the MSK Impact original panel that has 341 genes. Since then, over 505 genes have been incorporated. So maybe some of those additional genes have are especially predictive towards gallbladder rather than the bigger cancers, which is what the original one was for. So incorporating that signal might be helpful.
The other thing I might say is that we're thinking about ways in which we can reanalyze the MSK Impact data to give us information about other features that tend to be important, like mutation signature exposure and regional mutation density. And so maybe that'll help with the rarer cancers. That's where I think that work will be most useful. Thanks.
Great talk and great projects. My question is about the first project. You mentioned the importance of good calibration in medical settings and mentioned that this ensemble model was well calibrated out of the box. You got it for free. My question is, first, why do you think this particular model was well calibrated out of the box? And second, if it hadn't been, what might you have tried to improve it so that it was better calibrated?
Yeah, great questions. So first of all, multilayer perceptrons, especially in this case, are trained using cross-entropy error. And so with cross-entropy error, the perfect calibration is actually is the optimal of that error function. But like a lot of other things, like the way that random forests are trained, is not trained necessarily with this type of thing. So there's a word that if I had more sleep, I could tell you, but there's a certain type of function, objective function that ensures calibration. Now, the problem with multi-layer perceptrons is when you get data that's kind of out of distribution, that's very far away from the data sets that you've seen before, they have this tendency to be random, but super confident and wrong. But if you have an ensemble of them and they're random and wrong, they tend to be random and wrong in different ways. So that just reduces your confidence. So there's like two factors that are going on there, both the cross-entropy error and the fact that we're using ensemble to deal with these kind of out of distribution cases. Yeah, there are a variety of techniques to recalibrate. Yeah, and that's a whole field, which I would rather not go into because it's complicated. And where this is easy and it works out of the box.
Great, thank you very much.